Hi, my name is Christian Nansen. I'm from UC Davis, and this presentation is, as stated here, is about use of robotic rail imaging system in industry trials. I'm going to talk about towards the end how we're using optical sensing technology in our studies of plants. But I want to really talk more about how something that has existed for a long time, industry trials, the collaboration between universities and companies and IR4 and other partners, how that relationship, how valuable it is also from an academic standpoint, how we can use this type of collaboration with industry partners to prepare our students, how we can use it to discuss evolutionary biology, how we can use it in our teaching, how we can use it as a way not only to generate funds for our research and also publish it, but also use it to prepare our students in a way so that they can go out and have successful careers in the private industry. for somebody trying to become an applied entomologist or work in pest management or IPM in general, this should be a requirement to set up experiments like this. Even though the students themselves are not going to spray pesticides, having them read a label, have them understand the different parts of a label is incredibly valuable. Have them understand how to use pesticides properly, how to get rid of excess pesticides, how to use personal protection equipment, how to calibrate equipment. All the different calculations that go into this, I think, are incredibly valuable. Not only the proper use and the safe use of the equipment, but essentially come up, because there is this sort of notion out there that use of insecticides is kind of easy. All you have to do is spray. And we all know, us who actually do it on a regular basis, we all know that that's not the case. I mean, I'm wearing full on personal protection equipment here, and you have to turn off the fans in the greenhouse while you are spraying. So staying in a greenhouse for 45 minutes or longer, fully suited up like this, it is very, very hot. And I want to point out that there are some very good resources out there that you can potentially look into. They can be used in lab sessions for students actually to walk through some of these, learn more about use of pesticides, use of equipment, and particularly the calculations of how to find out how you actually figure out how much needs to be sprayed according to labeled rates. It's very important to learn about how we can spray, how this is being done properly with the right nozzles, with the right spray volume, with the right technology. How do you check pressure and all these things? So these are sort of basic aspects. And even though this sounds more sort of very much like bioengineering or something, well, there are actually ways, once you dig a little bit into it, that you can use it as a way to also talk about spray coverage and talk about how weather impacts the spray coverage. So now there's some modeling, some prediction of how spray coverages will be in certain canopies. So it starts getting more and more academic and more sort of geared towards math and modeling and predictions. And that can be taken even further if it's also associated with use of these water sensitive spray cards, which you see here on the screen here, where we have a little yellow card that turns blue. So I run lab sessions with my students where we actually experimentally test some of this. As you can see here, students are playing with fans and measuring the wind speed at different distances from the plants, and then afterwards using squirt bottles as a way to simulate spray applications. They also spray from different heights. So we have water sensitive spray cards placed in different parts of the canopy. So this is essentially to give students an appreciation for how easy, how little it takes to get an incomplete spray coverage. And we have also sort of taken that a couple of steps further. And this is where you then sort of, again, this is all based on industry trials that I've been doing over the years, right? So here I'm showing an example of a phone app we just published an article about. It's called Smart Spray. And together with undergraduate computer science students, we ended up developing this phone app here. So they got fully involved in this and actually became co-authors on this article we just published. And we now have Smart Spray as a tool that we're giving away for free, that people can use freely, both on iOS and Android. And we also took it one step further, which was we published an article at a website that is sort of targeting high school students. So we use this as an example of telling a story about how undergraduate students from computer science got interested in agriculture. So what I'm trying to show you here is there's so many ways that we can take a conventional industry trial and really use it in so many ways in our teaching. And we can sort of, if we sort of start focusing on spray coverage and others in addition to our treatments, we can then go beyond just counting the dead bugs or looking for phytotoxicity, because that in today's world is very difficult to publish. And it's also we can't have graduate theses sort of solely based on that. We need to add other response variables. We need to have other sides of this story to be added to it. So some of them that can be very easily included if we run it over a longer period of time is the plant morphometrics, basically the size of the plant, right? So the number of leaves or the height of the plant or something. So are they as a response to these treatments, and maybe indirectly because there are less pests on them, are the plants that were treated, are they bigger? Are they healthier looking? Then do they have more flowers than the plants that were in the control groups, right? In the negative control group. We can also do biochemical analysis of the plants themselves. Look at phytohormones. We can look at their element composition. So this is where we really start sort of looking at not only the potential mechanistic explanations for phytotoxicity and or for plants, how they respond to the bugs, but this is also trying to understand more about the plant physiology and linking that to the original treatments that we were looking at. And then we can, of course, if we are focusing on spray coverage, we could do some interesting studies where we actually look at avoidance, whether the pests are able to sense and then avoid. So this is particularly if spray coverage is not complete. So that then becomes a very sort of interesting behavioral study. And finally, and that's of course what I'm going to be talking about here, this is sort of supposed to be the focus of this talk, is also how leaf reflectance can be used as an indicator of not only how the plants are being affected by the stressor, but also maybe how they are recovering and how they're responding once they have been treated with the different treatments. If it's a systemic insecticide, so this could be plants even without pests, they could be treated with a systemic insecticides and then see how they are different from the negative controls. So this is where we start really sort of getting into explaining much more about how the chemicals, the insecticides, the treatments, how they are performing. And we could even look at leaf reflectance from different parts of the canopy if it's larger leaves. So we could sort of talk about a movement in a particular direction. So you can see hopefully that there are many, many ways we can take something rather trivial, which is just spray and then count. Well, we can build on that story, use it as a skeleton to go much further and gain much more knowledge about what is actually happening. And in that process, really enable students to learn a lot about commercial products and also about what is needed for us to push the agricultural development in the right direction and come up with more sustainable and effective pest management solutions in the future. And that allows us then to come up with something like this, which we recently published, right? So this was actually a study that was basically based on an industry trial we were doing, where we had chrysanthemum and gerbera plants, and we looked at and we tested this sort of two-part assumption, where we suspect that once arthropods infest a plant, we are expecting them to cause a significant change in the plant composition. And we were looking at the sort of the basic elements of the plants. So we could look at that, and we could demonstrate that there was an effect, and we tested different arthropods to see if they caused a unique plant response in terms of element composition. Because if that happens, then we would assume that the second part of the assumption also holds true, which is that if there's a significant change in the element composition, we would also expect the plants to reflect light differently and uniquely respond in terms of reflectance to the initial arthropod, the causal agent of the level of biotic stress. In the Nansen lab, we work with optical sensing data in multiple systems. One of them is we can take the plants directly out of the greenhouse into a dark room where we can control lighting conditions and image each of the plants individually. Here are two examples of plants being imaged inside under controlled conditions. And it's clear that the plants are very, very difficult to distinguish based on visual inspection. But if you actually look at the hyperspectral profiles from these plants, we can see here that there's actually some significant difference in the plant, especially out in the near infrared part of the spectrum, which is after about 700 nanometers. It is this difference, it is the hidden information in these reflectance profiles from plants that we use to classify plants and distinguish plants that have been under different stress levels. In addition to acquiring optical sensing data from individual plants that have been grown in cages and then being imaged inside a dark room under highly experimental conditions, we also in the Nansen lab have the capabilities outside. We have access to land and we have constructed hoop houses so we can grow plants under conditions that are more relevant to the commercial world. And we have developed technologies that allow us to collect optical sensing data during the day, but also at night with an active light source. Finally, in the Nansen lab, we also have the capability of mounting our optical sensing technologies on a drone so that we can collect data either from field plots or from plants that are on benches inside hoop houses after we have removed the top layer. So this is a way to work on a different spatial scale and combine it with all the other types of data that we can collect through our process of experimentation and manipulation of both plants and the environment in which they grow. In the Nansen lab at UC Davis, development and implementation of optical sensing solutions is shared with the public through our website.